the anatomy in a good amount of detail. We even talked about the histological staining patterns that we saw. And so as we move on to speak about the actual mechanism of muscle contraction, it's going to be important for you to recall that anatomy, for you to recall the different staining patterns, such as the I band and the A band, as well as be able to recall the overall anatomy of the myofibril and those two overlapping filaments. So muscle contraction is basically the shortening of the sarcomere. And because that is the most microscopic unit of muscle, if the sarcomere shortens, that'll bring about shortening of the myofibril, shortening of the muscle fiber itself, right, that cellular unit, as well as the entire muscle body. And this happens as the thick and thin filaments drag one another, right, specifically the thick dragging the thin towards the center of the sarcomere. So early physiologists used to think that there was actual contraction of the filaments themselves. And as we looked at this further with the development of the microscope and as we began to understand muscle a little bit better, what we realized is that neither of those filaments actually shorten themselves. What's happening is that the thick filament is dragging the thin filament. And that is what we call the sliding filament model. This theory of muscle physiology, which suggests that the thick filament latches onto the thin filament and drags it closer to the M lines, which are the center of the sarcomere, and that is what brings about the shortening of this entire thing and the shortening of muscle altogether. Okay, so as I mentioned here, we're only going to speak about crossbridge cycling, which is exactly how that force is generated, and then excitation-contraction coupling, how we can regulate, basically turn on and turn off muscle contraction. We will not speak about muscle metabolism. So here's what the histological staining pattern looks like in terms of the sliding filament model. What we see is that as the muscle contracts, the A band stays the same length. Okay, that's an important distinction because it's the only band, it's the only histological staining region that remains the same in length as the muscle shortens. And that is because the A band is the area of overlap. So as we can see, the area of overlap remains the same because both filaments are sliding in opposite directions, right? Really important to point that out. So the A band here, as muscle is relaxed, we can see it's the same width as the A band down here when the muscle is contracted. So because this is sliding, the thick filament is sliding to the outer periphery of the sarcomere, and because the thin filament in blue is being dragged to the inner periphery, not the inner periphery, but the center of the sarcomere, then that area of overlap essentially stays the same. The I band shortens, right? Because again, we're dragging the thin filaments towards the center. So the I bands are going to be shorter, as we can see here. The H zone shortens, right? We're dragging the thin filaments towards the middle of the sarcomere. And the H zone is the center of the sarcomere, which is represented by only thick filaments. So that region becomes much smaller as the thin filaments slide towards the center. And the entire sarcomere shortens, excepting for the A band, okay? Now this is brought about due to the cyclical formation and breaking of crossbridge cycles. And crossbridge cycles are the interaction between the thick filament, specifically the heads of these myosin filaments that are sticking up, and the thin filaments, and that interaction forms a crossbridge cycle, and that is made and broken over and over again in a cyclical pattern until the entire sarcomere shortens. Okay, and that will make a little bit more sense as we describe it a little better here. So here's an actual photomicrograph showing those staining patterns and showing it in a relaxed piece of muscle up here and then a contracted piece of muscle down here. So once again, we see the I bands, which are these lighter staining regions, becoming much shorter as the actin filaments drag towards the center. We see the H zone also becoming much smaller right in the middle. But the A band, which is a sort of moderately stained region, becomes, or stays the same rather, right? The A band remains the exact same in the relaxed form and in the contracted form. All right, let's get into the details of how these cross bridges are actually formed and broken and how we get this cyclical process that generates muscle contraction. So we have this link between actin and myosin, right, where actin is dragged towards the center of the sarcomere. And that happens because of the myosin head, which is reaching up and grabbing on. Now, the myosin head is going to undergo a conformational change. It's going to have two states. A high energy form, this is when it has ADP and an inorganic phosphate bound to it. Right? Remember, we talked about the two sites on that myosin head, one for binding actin, one for binding ADP. And so when it has ADP and that phosphate bound to it, it's in its high energy form, and it now has a higher affinity for actin. So it's going to be really attracted to actin. It's going to want to bind, make contact, and that is how we form that cross-bridge cycling. Now, in its low energy state, this is when it has ATP bound to it. So not in the hydrolyzed state, we just got ATP bound. And this is when it has a lower affinity for actin, and it's going to want to lose contact or break away from actin. And that is what is going to break that cross-bridge cycling. So we got to understand here that myosin is going to have a state where it's really attracted to actin. It's going to reach up and grab onto actin. And then it's going to have a low energy state where it's not attracted. It's going to let go of that bond. And that keeps happening over and over again, right? Making and breaking that contact. And that is what is essentially dragging the actin closer towards the center of the sarcomere. So this relies on the hydrolysis of ATP in order to liberate energy to actually drive this process. The hydrolysis of that ATP, which gives us that inorganic phosphate, that phosphate is holding the energy that is eventually going to do the dragging of the actin towards the M line. Right? Okay, now one way to kind of make an analogy that kind of makes this make sense is thinking about rowing a boat on water. So we're going to compare an oar paddle, right? If you think about an oar paddle that you would dip into the water and roll, right? Moving some water behind you as you propel the boat forward. We can kind of think of cross-bridge cycling in a similar fashion. Right? The oar paddle is what's making contact and breaking contact. And we can think about the thick and thin filament as when the oar paddle contacts the water, right? That is the breaking and making of that cross bridge. And so in that analogy, we can speak about the power stroke. So the power stroke is when the myosin head drags the actin towards the M line, and that is what propels the entire sarcomere to shorten, right? And so that is what, in our analogy, is what's going to move the boat forward. But in the instance of muscle, this is what is bringing the actin filament closer to the M line and shortening the entire sarcomere. So when the thick and thin filaments detach, that is when the oar breaks contact, and we can move our oar to a new position in order to drag more water behind us. Similarly, we're going to move the myosin head to a new position on actin to drag that thin filament even closer towards the M line. Okay, and so the myosin head is going to reposition itself to a new spot to bring that actin closer. I hope that analogy kind of makes sense. It's one that usually kind of gives you an idea of what this looks like as we speak about these microscopic structures in a real way, right? So we've got this myosin head reaching up, grabbing onto actin, dragging it close to the M line. It's going to reposition itself, grab another bit, and drag it closer again. And that happens over and over again until the sarcomere is completely shortened or the muscle is completely contracted. Okay, and so we can look at this schematic as representing the steps in that process. So let's go through these steps one by one as we speak about how cross-bridge cycling is actually brought about. So to start, let's remind ourselves of the structure of these filamentous proteins, right? We've got actin here in blue, and actin is nothing but these different monomers that are strung together like pearls on a string, we describe them. And then we've also got myosin, and myosin has these heads that are sticking up that have the two binding spots, one binding spot for that ADP and that phosphate, and the other binding site for actin, right? Whereas on our thin filament, we have the binding spot for myosin, which are these black dots. We've got this regulatory protein, troponin, and this other protein, tropomyosin. And so these two proteins are going to be important in the regulation of this process. But for now, we're really going to focus on looking at where myosin makes contact with actin. Okay. So we start off with myosin in its high energy state. Remember we said when it has ADP and that inorganic phosphate bound to it, it's in a state where it's really attracted to actin. And so as we can see here, it's made contact with actin because it's in that high energy form. Subsequently, the phosphate is released off, right? We release that inorganic phosphate off, and that is what liberates the energy to complete the next step, which is the power stroke. So the power stroke is the actual dragging of that actin filament closer towards the center of the sarcomere or towards the M line, right? So the phosphate that is released off liberates the energy to drag actin towards the M line, and we call this second step the power stroke. Now the ADP is released off, so after the ADP comes off, now we still have actin bound to myosin. And the reason we still have actin bound to myosin is because we don't have ATP on there yet. We talked about this myosin head being in two states. The high energy state is when it has this ADP and this inorganic phosphate, but the low energy state is when it has ATP bound. And until we get a new ATP molecule here, this myosin head is going to remain tightly bound to this actin filament, okay? And this is where we get the concept of rigor. So rigor is this state where myosin is still in its low energy form. Although we don't have a new ATP in here, we need that ATP to come in order for myosin to let go of actin. And so this is what brings about rigor mortis. If you've ever heard of rigor mortis, which is the state where your muscles stay tightly contracted after death, and that is because in the absence of ATP, right, after you die, there's no ATP circulating. And so myosin stays tightly bound to actin, and essentially that muscle is going to stay tightly contracted in the state that it was in. And that's why even after death, a person remains or a cadaver remains in the exact same state that it was if rigor mortis is allowed to set in. And this is the physiologic explanation for that rigor mortis. Now the next step is a new ATP comes in and binds onto that myosin head, and that will now unbind myosin from actin. So a new ATP must come in so that the myosin head can go back into its low energy state where it is not attracted to actin, and it's going to let go of that actin filament, okay? And that is what breaks the contact of actin and myosin. Now that second ATP, that new ATP is hydrolyzed into ADP and inorganic phosphate once again, and that prepares myosin, the myosin head, to be in its high energy state once again, and it's going to become attracted to actin and reach up and grab onto that actin. And that's where we describe the myosin head as being in its cocked state, where it's ready to make contact with actin once again. And this process happens over and over and over again until that actin is brought to the center of the sarcomere, and that is when the muscle is fully contracted, right? So it doesn't just happen once, but it's happening again and again and again and again until we have full contraction or full shortening of the sarcomere, okay? And so these are the 5 steps that are important for crossbridge cycling. The binding of myosin to actin, the power stroke, which is the liberating of that inorganic phosphate, and the dragging of actin. We've got rigor, which is the third step, that tight interaction between actin and myosin until a new ATP comes in. We've got the unbinding of myosin when that new ATP comes in. And then we've got the hydrolysis of that new ATP, which brings the myosin head back in its cocked position. Alrighty, okay. Now, I'm going to break and see if there are any questions about that. And in the meantime, while I kind of wait to see if there are any questions coming up, there's a really nice video that I've posted in Blackboard. If you haven't looked at it already, I suggest that you kind of look at it after this. And it's going to be a good way to summarize all of these steps in a 3D way that will make you appreciate them better. So have a look at this short video if you haven't already done so. And that's going to kind of delineate those 5 steps in a more 3D realistic way. Okay, so I don't see any questions popping up, so we're going to proceed right ahead. And here we're going to look at excitation-contraction coupling. So this is the second important process for muscle contraction. This is what regulates muscle contraction. This is what turns it on and turns it off. So as you would imagine, the body being this highly efficient structures, right? This highly efficient system, we don't have muscles contracting indefinitely, and we don't have this process happening all the time. And so in order to regulate this process, calcium is going to play that role of turning contractions on, and then the absence of calcium is going to play the role of turning contractions off, okay? And so here we're going to speak about the sequence of events where an action potential, and if you remember from our last unit, we described this to be an end plate potential, where this action potential in the sarcolemma causes contraction. So how do we connect the dots from the events that happen on the neuromuscular junction where that end plate potential is brought about to the actual shortening of the sarcomere to the crossbridge cycling? How do we fill in the steps of that communication? And that's what excitation-contraction coupling is. So this is going to depend on neural input at the neuromuscular junction coming from the motor neuron. That's going to result in release of our neurotransmitter, which is acetylcholine. And that requires calcium release from the sarcoplasmic reticulum, which is then important in interacting with the regulatory proteins on actin and turning on this muscle contraction. Okay. So this slide is really a review of what we spoke about in terms of the events at the neuromuscular junction, the somatic nervous system. So we know that each motor neuron innervates several muscle fibers, and we also know that each muscle fiber receives neural input from only one motor neuron. And so this gave us the understanding of the motor unit, right? One motor neuron and all of the fibers that it was innervating. And so this is similar to an ordinary synapse in that acetylcholine is going to be released from the axon terminal, and that is going to bring about an end plate potential on the motor end plate. And if you remember, we described the motor end plate as being that highly folded membrane of the skeletal muscle fiber. And it's highly folded in order to increase the surface area, which will help to spread that end plate potential and transition or convert that electric energy, that end plate potential, throughout the entire muscle fiber and bring about contraction. Okay. So let's go on to speak about the role of calcium in all of this. So if you remember the two regulatory proteins, troponin and tropomyosin, that are interacting with actin, this is where calcium comes into the picture. So troponin is going to have a binding place for calcium. And if there is no calcium bound to troponin, tropomyosin is going to cover up the binding spots on actin and essentially prevent myosin from binding onto actin. Okay, I'll say that again. So if there's no calcium bound onto troponin, remember troponin has a binding spot for actin, for tropomyosin, as well as for calcium. So if there's no calcium on that calcium spot, then tropomyosin is going to cover up those binding spots, and we cannot have crossbridge cycling because actin and myosin are not going to be able to interact. In this state, muscle is relaxed and we do not have contraction. Now, if calcium is present, it's going to bind onto that binding spot on troponin, and it's going to slide tropomyosin off of those actin, off of those myosin binding spots on actin, and that is going to uncover those binding spots and allow myosin to reach up and bind onto those binding spots, essentially forming that crossbridge cycling, and in that state, muscle will begin to contract. So this is how calcium regulates this process. If there's no calcium, muscle contraction is turned off and the muscle is in its relaxed state. And in the presence of calcium, muscle contraction is turned on and the muscle is in its contracted state. So here are the steps that govern or bring about that excitation-contraction coupling. And the first one is going to be the action potential generated in the sarcolemma. So that's really going to pick us up where we left off, describing the events of the neuromuscular junction. We bring that neurotransmitter across the cleft, it binds onto those receptors, those nicotinic receptors, and we generate an action potential, which we described as an end plate potential, and that is propagated throughout the muscle with the help of the T-tubules. So the T-tubules is how we propagate this action potential throughout the muscle. And then we've got these receptors that are on the sarcoplasmic reticulum that can couple the binding of calcium to that end plate potential. And so when we get that muscle when we get that end plate potential rather, running through the T-tubules, that is going to bring about the opening up of the channels on the sarcoplasmic reticulum such that calcium can flood the cytosol of the muscle fiber. It can bind onto troponin. It can shift tropomyosin off of those binding sites, and then cross-bridge cycling can be initiated. All right, so these are the 6 steps, and we're actually going to walk through this schematic image that kind of depicts these steps in more detail. So the first step is acetylcholine is released from the axon terminal of the neuromuscular junction here. It's going to cross the cleft and it's going to bind on to these nicotinic cholinergic receptors, and that is going to bring about an end plate potential at the motor end plate. This end plate potential or this action potential is going to propagate throughout the entire muscle body. It's going to travel along the T-tubules, which are these invaginations that we talked about. Remember, we described the triad as being the T-tubule in the center, and then the lateral sacs, which are formed by the sarcoplasmic reticulum. And so the T-tubules are these invaginations or these tunnels that help to spread the action potential throughout the muscle very quickly. The third step here is that the action potential is going to bind to receptors that are in the sarcoplasmic reticulum and trigger the release of calcium from the storage area, which is in the SR, out into the cytosol of the muscle fiber. So let's back up here a little bit. If we remember that the sarcoplasmic reticulum, which in muscle is the storage area for calcium, this is where calcium is being stored, and this is why we don't have muscle contracting all the time. The calcium that is important for regulating this process is actually stored away in the SR until the action potential turns these receptors on that actually open up these channels and bring this calcium out into the cytoplasm. Alrighty? And so when that calcium floods the cytoplasm, it now comes into contact with actin, specifically the binding site on troponin, which is this pink regulatory protein. Calcium can bind onto troponin, and then that can drag tropomyosin. So tropomyosin is this thread-like protein in purple. And once that gets dragged off of these myosin binding sites, then myosin can reach up and interact with these myosin binding sites on actin and start to bring about cross-bridge cycling. Now, after that is done, we must get rid of all the calcium that's in the cytosol. This is very important for ceasing muscle contraction. Otherwise, we would have muscle contracting indefinitely, which we do not want. So we must actively transport this calcium from the cytosol or the cytoplasm back into the sarcoplasmic reticulum, where it can be stored until another end plate potential or another action potential is brought about. And so once that happens, if there's no longer calcium present in the cytosol, then the troponin will not have calcium bound onto it. And so it's going to drag tropomyosin back to cover up those myosin binding sites once again. And that is going to bring about relaxation of the muscle, as actin and myosin can no longer come in contact with each other. Okay. Alrighty. So here is what actin looks like in its two states. In its relaxed state, we can see that troponin, which is one of these regulatory proteins associated with actin, does not have calcium bound onto it. And I keep describing this relationship here. Remember, troponin has three binding spots. It has a spot to bind calcium. It has a spot to bind tropomyosin, which is this thread-like regulatory protein, and it has a spot to bind the actual actin filament. And so when it doesn't have calcium on here, it's going to drag that tropomyosin to cover up these myosin binding sites. And so in this relaxed state, actin cannot interact with myosin because these myosin binding spots are covered up. In its contracted state, actin does not have these tropomyosin filaments covering up the binding sites for myosin. And that's because calcium binds onto its spot on troponin, and that drags this tropomyosin protein off of the binding sites, uncovering them so that the myosin head can reach up and make contact at these exposed myosin binding sites. So this is how we say that calcium regulates this process. If there's no calcium, actin cannot bind to myosin. We cannot have cross-bridge cycling or shortening or contracting of the muscle. If there is calcium, actin is now exposed, right? Those myosin heads on actin are now exposed. And so myosin can interact with actin and bring about that cross-bridge cycling or shortening of the sarcomere. All right, so I'm going to pause here once again and see what questions may come up. So that is essentially the crux, the important parts of excitation-contraction coupling. And again, there's another sort of short, concise video that will summarize these events. So these are the two important processes of understanding how muscle contraction is brought about. And just to summarize here, we talked about the steps of how the actual shortening comes about. And then we spoke about the steps of how this is regulated, right? We don't just have this running all the time, but we have very controlled instances of muscle contraction only when that action potential opens up those receptors and floods the system, floods the cytosol, sorry, with calcium. That calcium can go in, change the configuration of actin, and bring about that cross-bridge cycling. So we spoke about the actual shortening, and we spoke about the regulatory process of muscle contraction. Okay. All right. So this next slide demonstrates how that action potential is actually opening up these channels on the sarcoplasmic reticulum. So because of the fact that we have gating of the sarcoplasmic reticulum, calcium cannot just leave the SR as it pleases, right? Calcium is withheld within the sarcoplasmic reticulum, And there are certain gating mechanisms on the membrane of that SR that will then allow calcium to leave only when an action potential is propagated through this T-tubule. So we have this coupling of ryanodine receptors and DHP receptors, two receptors that are found studded on the membrane of the SR, as well as the plasma membrane or the sarcolemma itself. So this is the membrane of the muscle fiber, which we call the sarcolemma. This is the membrane of the sarcoplasmic reticulum where calcium is being stored. And so if we remind ourselves that calcium needs to move from the SR in blue out into the cytosol here in white in order to come in contact with that actin and to expose those binding sites. So the two receptors that are found in this membrane, they actually control the opening of this channel, which allows that calcium to open up and flood the cytosol. So the T-tubule, right, the T-tubule shown here has the DHP receptor embedded in it. And the sarcoplasmic reticulum membrane, I can never say that word, sarcoplasmic reticulum membrane has the ryanodine receptor embedded into it. And these two receptors are coupled to each other, meaning that they're touching each other and they're communicating. So when the action potential propagates down the T-tubule, it's going to come in contact with the DHP receptor first as it goes down the T-tubule. And then that is going to signal the ryanodine receptor, which is touching the DHP receptor, to open up this channel. So the ryanodine receptor is not only a receptor, but it's also a calcium channel. So when that action potential comes in contact here, it couples to the opening of that calcium channel, which brings calcium from the stored region here in the SR out into the cytosol, where it can come in contact with actin and actually start turning on muscle contraction. So this is how these two proteins are also involved in the regulation of this process. Okay. And as we talked about, in order for termination of contraction to be brought about, calcium must leave troponin and it must go back into the sarcoplasmic reticulum. It must leave the cytosol, and it is done so actively. So there are active calcium ATPase pumps that are on the sarcoplasmic reticulum that is pumping this calcium back into the SR, and that is what is ceasing muscle contraction as actin goes back into its relaxed state, where those tropomyosin filaments are going to cover up the myosin binding sites once again.